Hello, my name is Adam Goodman, and I run the PGT department for NextGen MDX, a US-based genetics lab located in Grand Rapids, Michigan. At NextGen MDX, we make it our mission to ensure our patients have access to best-in-class science, unrivaled accuracy, and personal genetic counseling designed to help them chart a course for their family's future. To that end, we offer our clients state-of-the-art carrier screening, pre-implantation genetic testing, non-invasive pregnancy testing, hereditary cancer screening, molecular vaginosis testing, and more recently, we have become the largest COVID-19 testing laboratory in the state of Michigan, having run nearly 1 million tests since March 2020. Today, I'll be focusing on the platform we use for PGTA and why it has become our assay of choice. The goal of PGTA as a science is to identify those embryos with the greatest potential of resulting in a healthy pregnancy and baby. To accomplish this goal, a good PGTA assay must correctly identify aneuploid embryos. At the same time, we must be able to be sure that the aneuploidy events that are being detected really exist. If an assay does not provide tools to properly investigate sample and data quality, it can be prone to both false positives and false negatives. And finally, cost. I would be remiss if I did not mention that cost was a big factor. The PGTA field has become extremely competitive over the last decade. Many of our IVF lab clients are big believers in improving their success rates with PGT, and lower cost means greater patient utilization. As such, small technological improvements that come with a significant price hike are often ill-received in the field. If patients cannot afford the test, it does little good. Here are the fundamental areas I like to investigate when evaluating a PGTA assay. Resolution and accuracy. I want to know the assay provides high-resolution 24-chromosome data with the ability to reliably identify mosaicism. We need an assay that has a resolution high enough to accurately detect not just whole chromosome gains and losses, but also smaller segmental deletions and duplications, as numerous research studies have shown that they too can be responsible for reduced implantation and increased miscarriage rates. Reliability and precision. I believe using isolated genomic DNA to validate a PGTA assay is too easy. You're going to get great quality data every time because you have pristine quality input samples. I am more interested in testing out real-world analogs of variable quality because in practice, not all samples received at a reference lab are going to be perfect. Data interpretation. Does the assay provide strong software capable of allowing our lab to thoroughly investigate samples, not just for abnormalities, but also for assessing the quality of the data itself, so that we can better separate sample noise and amplification artifacts from true aberrations? Throughput and ease of use. How long does the assay take to run? How much of that is hands-on? And how amenable is the assay to automation? And finally, I want to know more about the associated cost than just the per sample price under ideal circumstances. I want to know the per run cost and the run size options available. Very few labs have enough sample volume to fill every run to its maximum, so you need to know what your costs will be for runs performed under 100% capacity. Labor costs are also another important factor. What level of experience is required and for how long at the various stages of sample handling. Assays that use more automation can free up senior technician time without reducing quality. Additionally, there can be significant differences in ancillary costs, such as plastic consumables and safety equipment. The vast majority of all PGTA is performed utilizing either Thermo Fisher's ReproSeq PGS assay or Vitrolife's VeriSeq PGS assay, originally developed by Illumina. There are a couple of other options out there. However, assuming you agree, the best way to perform PGTA, as most scientists in proprietary sequencer reagents. These sequencing reagents comprise the bulk of the PGTA reagent cost. Therefore, all third-party options that I have explored have been cost prohibitive, as no matter how low the cost of their library prep reagents, one must still purchase the sequencing reagents from Thermo Fisher or Illumina. Is only financially feasible if resolution is severely sacrificed. Be on the lookout for new technologies to advance our program. We were still using FISH and PCR-based tests to assess chromosomal composition in day 3 embryo biopsies. And I've been able to experience firsthand the evolution of PGT through many different platforms. When VeriSeq PGS first came to market, it revolutionized the PGTA field, drastically increasing both quality and throughput. I spent nearly a decade working with the VeriSeq assay, and I still think it is a good product. However, I am always open to new ideas, and when the latest version of Thermo's represent for the same level of high-resolution sequencing as VeriSeq, but with an easier protocol and lower price. Since at NextGen we employ both Illumina and Thermo Fisher sequencers on a large scale in other divisions, we are fortunate in that we can easily switch between sequencing platforms for PGTA. And we decided to give the ReproSeq assay a try to see if it would meet our high standards. Resolution. For our investigation of resolution, we purchased 10 different Coriell cell lines with known abnormalities ranging in size from whole chromosome down to 1.5 megabase pairs. One important distinction here is that we tested actual intact cells, not isolated DNA. Again, I believe this approach provides a better analog of real-world performance with regard to embryo biopsies. We diluted cell suspensions down to the amount found in typical PGT biopsies, aliquoting approximately 5 to 10 cells per collection tube. We then ran them multiple times through both the VeriSeq and ReproSeq platforms to see if we could empirically find any differences in their limit of detection with regards to the identification of segmental deletions and duplications. We found that both assays outperformed their manufacturer's stated resolutions of 10 megabase pairs, reliably detecting segmental changes down to about 5 megabase pairs before we started to see any missed calls. Both assays showed identical results for all samples, not just in the limits of detection, but all the corresponding copy number charts were virtually perfect overlays of each other. For our assessment of accuracy, we again wanted to test an analog more rigorous than isolated DNA, so we partnered with an IVF lab in Florida to retest embryos that were previously predicted to be aneuploid by a third party using a SNP-based array platform. Since the experiment involved running the samples simultaneously through both the VeriSeq and ReproSeq assays, this was helpful in that it could potentially provide a tiebreaker in the event of a discrepancy between the two NGS-based platforms. Additionally, we tested not just a trophectoderm biopsy from these samples, but also the remainder of the whole embryos themselves were tested to assess concurrence between biopsy and embryo calls. After all, the whole point of PGTA is to be able to accurately predict the ploidy of the embryos. 22 embryos yielding 44 unique samples made it into the study. NextGen was blind to the third-party aneuploidy predictions, which samples tubes contained biopsies, which contained whole embryos, and also to which embryo a given biopsy belonged. We found 100% concordance between the ReproSeq and VeriSeq platforms. At the fundamental level of calling a sample normal or abnormal, we also saw 100% concordance between the biopsies and their corresponding whole embryo sample. However, we did see 2 instances in which the abnormalities detected on the biopsy did not perfectly match that seen in the whole embryo sample. Those sample sets are seen here in red, and they are not necessarily surprising in that the abnormalities detected are related, even though they are not perfect matches. Other studies have shown that with mosaic samples, we sometimes see variations of the same abnormality when a sample is biopsied multiple times, such as a trisomy in one biopsy and a reciprocal monosomy in another biopsy from the same embryo. The more surprising discovery we made was that 4 of the embryos previously predicted to be abnormal by third-party SNP array were found to be euploid. In these instances, both the biopsy and the whole embryo samples were found to be euploid on both ReproSeq and VeriSeq platforms. These samples are highlighted in red. In my opinion, I believe the most plausible explanation is erroneous identification of sample noise as an aneuploidy event during SNP assay testing. There were 3 other discrepancies between the third-party SNP testing and our 2 NGS-based assays seen in blue. However, in these instances, the abnormalities seen were related and likely implicate technical limitations of the SNP assay and/or embryo mosaicism as the cause of the discordance. Next, we sought to test the reliability and precision of the ReproSeq assay. First, all samples from the resolution study were run a second time to ensure no call changes were seen. None were. Then we filled an entire run using only one sample 24 times. This sample had a known 40% mosaic trisomy X karyotype and was used to check for consistency in reads per sample, as well as percent mosaicism detected by the ReproSeq assay. We averaged over 145,000 reads per sample after filtering, with optimal being defined by the manufacturer as anything above 100,000 reads per sample. Additionally, we saw all samples stay fairly close to the expected 40% mosaic value, with a mean level of 39%. The entire experiment was then repeated by a second technician to verify reproducibility of these numbers in a second set of hands. Overall, the coefficient of variation for reads after filtering was less than 10% for both technicians, indicating great evenness of sample representation across the entire ReproSeq sequencing chip. With regards to mosaicism, we saw plus or minus 10% from the expected value of 40%. This is spot on what I have seen over the years with the VeriSeq assay through thousands of samples tested and represents what I believe to be the current best available precision for a commercial PGTA assay. However, it also represents one of the reasons why I do not think it is a good idea to stratify mosaicism into low-level and high-level groupings on PGTA reports. As you can see, even under ideal circumstances, the exact same sample could show up as 30% mosaic in one instance and 50% mosaic in another, a difference of 20%, even within the very same sequencing run. Ultimately, we found the scientific performance of these two assays to be virtually identical. So in deciding which assay to integrate into our laboratory, the most heavily weighted factors became protocol ease of use, throughput, scalability and flexibility, software performance, and cost. One of the biggest differences between ReproSeq and VeriSeq protocols is the point at which samples can be pooled. In the ReproSeq protocol, because barcoding occurs as a part of the whole genome amplification process, samples are pooled immediately following amplification. This has huge implications on the number of pipetting steps and plasticware used downstream. It makes the ReproSeq workflow much simpler and results in far less labware being used compared to the VeriSeq protocol, where samples cannot be pooled until right before loading the sequencer. After whole genome amplification, the ReproSeq protocol essentially uses the same small amount of plasticware whether processing 1 or 96 samples. With 96 samples in the VeriSeq workflow, you use at least 96 times more tips, plates, purification beads, etc. These pictures show the actual amount of consumables required to take 96 samples from post-amplification to ready-to-load sequencing chip/flow cell on the two different assays. On the left, 3 racks of tips and a few microcentrifuge tubes are more than enough with ReproSeq, and on the right, VeriSeq uses 27 racks of tips, 6 PCR plates, 2 deep well plates, 8 reagent reservoirs, a handful of assorted conical tubes, and repeater pipette tips. Of note, one would utilize even more standard tips without the use of the repeater pipette. Although both ReproSeq and VeriSeq use magnetic bead PCR cleanup protocols that are fundamentally the same, they are very different in practice. Since ReproSeq samples are pooled right after amplification, the magnetic bead cleanup stage is very easy, whether working with 1 or 96 samples. The protocol involves a single reaction in a single tube. On the other hand, the VeriSeq protocol requires a separate cleanup reaction for each individual sample. This means that a multi-channel pipetter must be used in conjunction with a deep well plate on a magnetic stand, and as anyone who has ever performed such a deep well cleanup can attest, This is a very delicate procedure. If you accidentally aspirate the magnetic beads at the wrong time, you can drastically compromise the sample's representation on a sequencing run. Accordingly, the VeriSeq cleanup involves more time, more pipetting steps, and consumes far more tips. Additionally, the VeriSeq protocol uses 96 times more magnetic beads, which are pricey and not included in either assay's kit. Both ReproSeq and VeriSeq protocols can be done inside 12 hours, but more practically, they are split over 2 workdays. The ReproSeq protocol can utilize 3 different sequencing chips, allowing for run sizes of 16, 24, or 96 samples, while the VeriSeq protocol has only 1 chip size intended to run 24 samples at a time. Whole genome amplification for both ReproSeq and VeriSeq use the same underlying chemistry, and the WGA workflow is essentially identical with the exception of adding barcode indices in the 3rd stage of the WGA process with the ReproSeq assay, which allows for pooling of the samples prior to template preparation. I find the biggest difference between the 2 assays is ReproSeq's plug-and-play automation of the lengthy and sensitive template preparation chemistry needed prior to sequencing. This is accomplished via Thermo's Ion Chef robot. It essentially frees up 5 hours of tech time compared to the manual VeriSeq protocol. Additionally, the Ion Chef gives you great flexibility in that it allows you to dictate precisely when you would like the robot to finish. You can tell it to be done in as fast as 5 hours, regardless of the run size, or you can tell it to finish up at, say, 9 a.m. the following morning, so the sequencing chip is ready to load on the sequencer at the beginning of the next workday. You can see here that the overall protocol length is very similar for these 2 assays at the 24 sample per run level. However, ReproSeq does have a scaling advantage as VeriSeq is limited to 24 sample sequencing run capacity. With VeriSeq, each additional set of 24 samples will add 3 hours of sequencing time, plus at least an hour of extra prep time, and that is assuming all samples are prepped simultaneously. If prepped separately, the total time doubles. With regards to the hands-on time reported by manufacturers, you must be careful to note the duration and frequency of breaks between steps. For example, though the template preparation portion of the VeriSeq PGS protocol requires only 90 minutes of direct hands-on time, in practice our technicians are tied up for a full 5 hours, as the longest break in the protocol is just 35 minutes. I think the more practical metric here is uninterrupted hands-off time. Both assays afford the user about an hour of free time during the whole genome amplification, but the automated Ion Chef library prep frees up an extra 4.5 hours of usable tech time compared to the manual VeriSeq library prep. Both assays come with their own software, and both are fully capable of automatically identifying whole chromosome aneuploidy events based on variations in sequencing data. Thermo's Ion Reporter goes a step further and can automatically flag segmental and mosaic aneuploidy events, whereas Illumina's BlueFuse software can only make whole chromosome calls. With BlueFuse, it is still possible to make segmental and mosaic calls based on the CNV charts, but this must be done manually. The Ion Reporter software also includes coverage maps for the assessment of data quality, and it allows for the use of custom baselines should we ever need to use a different amplification method, say for example when using multiple displacement amplification for targeting a difficult to amplify single gene disorder for concurrent PGTM testing. With regards to pricing, our lab saw only a relatively modest reduction in reagent costs for ReproSeq PGS compared to VeriSeq PGS, about a 15% reduction at the 24 sample per run level. However, the ReproSeq workflow also uses far less consumable plasticware. Does this drastically lower the cost per sample? No, but it easily tacks on a couple hundred of US dollars per run, which adds up over time. Additionally, unlike VeriSeq, the ReproSeq assay does not utilize hazardous reagents, which means you do not need to invest in a fume hood and you do not need to pay for expensive hazardous waste disposal. Additionally, less hours of expert tech time are required with ReproSeq due to the simpler workflow and the Ion Chef robot's full automation of template preparation. Copy number assays inherently involve sensitive reactions that require experienced and expertly trained lab technicians to achieve high-quality results consistently. Being able to go from pool to ready to sequence chip on the Ion Chef allows us to run more samples with fewer expert technicians while simultaneously removing some human imprecision from the process. In summary, we were quite impressed with the ReproSeq PGS platform. Empirically, we could find nothing that VeriSeq could detect that ReproSeq could not, and we found the ReproSeq assay more reliable and much easier to use. The Ion Chef makes processing samples much simpler, and data interpretation is also easier on the more powerful Ion Reporter software. Combined with the reduction in costs, we found ReproSeq PGS to currently be the best assay for our lab and is now what we use on a daily basis at NextGen MDX. Thank you. And now I'm ready for any questions.